Good morning, everyone, and thank you for being here. Today, we are going to dive into a rapidly evolving and truly fascinating area of microbiology, the human microbiome. The title of today's session is Microbiome: Composition, Function, and Dysbiosis. This session will be particularly valuable for those of you interested in medical microbiology, immunology, human health, and translational research. Now, when we think about the human body, we often focus on the cells that are innately ours: epithelial cells, neurons, immune cells, and so on. But it turns out we are not alone in our own bodies. In fact, we're vastly outnumbered. We carry about 39 trillion microbial cells, bacteria, viruses, archaea, fungi, and only about 30 trillion human cells. Genetically, the disparity is even more dramatic. The collective genetic material of the microbiota, called the microbiome, is over 100 times larger than our own genome. So in many ways, we are superorganisms, human cells working alongside microbial collaborators. This body of microorganisms and their genes interact with us every single moment, affecting our digestion, our immunity, even our mood and behavior. We're going to cover the composition of the microbiota at various body sites, delve into the functions these microbial communities perform, and then explore dysbiosis, the breakdown of microbial balance, which is linked to a wide range of diseases from inflammatory bowel disease, IBD, to obesity, and even neurological disorders. The field of microbiome research is still relatively young, but the insights we've gained so far are transforming our understanding of health and disease and paving the way for novel treatments, including microbiota transplants, personalized probiotics, and nutritional therapeutics. So, let's begin our exploration. Let's start by defining the key terms: microbiota and microbiome. The microbiota refers to the community of microorganisms themselves, bacteria, fungi, viruses, archaea, that inhabit a particular environment. When we talk about the gut microbiota, we are referring to the actual living organisms residing in the gastrointestinal tract. On the other hand, the microbiome refers to the collective genomes of these microorganisms. Think of the microbiota as the actors on stage and the microbiome as the script they bring with them. Both are crucial for the performance. Historically, our understanding of microbes was centered around pathogens, those few species that cause disease. But with the advent of metagenomic sequencing, especially since the Human Microbiome Project launched in 2007, we began to uncover the vast, previously unseen microbial world that coexists with us in a largely symbiotic relationship. Let me give you a quote that often comes up in microbiome literature: "The microbiome is our second genome." This phrase encapsulates the idea that these microbes are not just passengers, but essential players in our biology. Why does this matter? Because microbes contribute critical functions that we cannot perform ourselves. For example, we cannot digest certain complex carbohydrates without the help of bacterial enzymes. We also rely on microbes to synthesize essential vitamins like K2 and certain B vitamins. They help develop our immune system and protect us from pathogens. So when we talk about health, we can no longer think of it purely in terms of our own cells. We must consider the entire ecosystem, including these microbial communities. That's the foundation we build on today. Each region of the human body is home to a unique microbial community adapted to its local environment. Let's start with the gut microbiota, the most diverse and dense, housing trillions of organisms dominated by firmicutes and bacteroidetes. These microbes digest fiber, produce vitamins, and modulate immunity. On the skin, microbial variation depends on moisture and oil content. Sebaceous areas favor Cutibacterium, while moist regions host Staphylococcus and Corynebacterium. These microbes defend against pathogens and educate immune cells. In the oral cavity, surfaces like the tongue, teeth, and gums support complex biofilms. Genera like Streptococcus, Veillonella, and Actinomyces dominate. Constant exposure to food and saliva keeps this ecosystem dynamic. The respiratory tract, especially the nose and throat, hosts Haemophilus, Neisseria, and Staphylococcus. The lungs, once thought sterile, also harbor low biomass communities that influence immunity. The urogenital tract varies by sex. In females, lactobacillus species maintain acidic pH in the vagina, preventing infections. In males, microbiota resembles that of the skin and urinary tract. Even the eye hosts microbes like Staphylococcus, which help prevent infection. These communities don't exist in isolation. Crosstalk exists, for example, between the gut and skin or brain. Disruption in one site can ripple systemically. Understanding this site-specific colonization is foundational for exploring health and disease. The gut microbiota is the largest and most complex human microbial ecosystem. It resides mainly in the colon and includes firmicutes, bacteroidetes, actinobacteria, and others. Each person harbors a unique microbial signature influenced by diet, age, genetics, and health. Functions are diverse and vital. Gut microbes ferment undigested carbohydrates into short-chain fatty acids, SCFAs, like butyrate, which nourish colon cells and reduce inflammation. They also synthesize vitamins such as K, B12, and folate. Microbes train the immune system from infancy and compete with pathogens to protect against infection, a process known as colonization resistance. Some even influence how drugs are metabolized, affecting their efficacy. Factors shaping the microbiota include birth method, antibiotics, diet, stress, and aging. Fiber-rich diets increase diversity, antibiotics and Western diets reduce it. Vaginal birth promotes a richer initial microbiota compared to cesarean delivery. Excitingly, the gut-brain axis shows that gut microbes can influence mood and behavior via metabolites and immune signaling. In summary, the gut microbiota is a key regulator of digestion, immunity, and overall health. When disrupted, what we call dysbiosis, the impact can be systemic and severe. The skin microbiota is the body's first microbial barrier, playing a vital role in immune defense and environmental sensing. While often overlooked, it's incredibly diverse and highly site-specific. The skin is composed of multiple microenvironments, sebaceous, oily, moist, and dry areas, all of which support distinct microbial populations. For example, sebaceous regions like the face and upper back are rich in Cutibacterium acnes, which thrives in lipid-rich environments and contributes to conditions like acne when dysregulated. Moist areas, such as the armpits and groin, favor Staphylococcus and Corynebacterium due to higher humidity and temperature. Dry areas, like the forearms and legs, tend to have more microbial diversity but lower density. Importantly, these microbes don't just sit passively on the skin. They produce antimicrobial peptides, compete with pathogens for space and nutrients, and interact with host immune cells to calibrate inflammatory responses. For instance, Staphylococcus epidermidis helps prevent colonization by Staphylococcus aureus, a common skin pathogen. The skin microbiota is shaped by numerous factors: age, hygiene, climate, genetics, and antibiotic use. Disruptions in this ecosystem, through overwashing, topical antimicrobials, or skin barrier dysfunction, can lead to dysbiosis, contributing to conditions like eczema, psoriasis, and wound infections. Understanding the skin microbiota helps not just in dermatology but in systemic health, especially when we consider how skin microbes interact with the immune system across the lifespan. The oral cavity hosts one of the most dynamic and diverse microbiotas in the body. It's a warm, moist, nutrient-rich environment that includes teeth, tongue, cheeks, and saliva, each supporting different communities. Key players include Streptococcus, Fusobacterium, Veillonella, and Actinomyces. Many of these microbes form structured biofilms, especially on teeth, plaque. These communities interact with one another and the host immune system. When balanced, they maintain oral health. But in dysbiosis, they can drive dental caries, periodontitis, and even systemic infections via translocation into the bloodstream. The upper respiratory tract, including the nasal passages, sinuses, and throat, share some overlap with the oral microbiota. Common residents include Corynebacterium, Staphylococcus, Haemophilus, and Neisseria. These microbes compete with pathogens like Streptococcus pneumoniae and Staphylococcus aureus, helping prevent infections. The lower respiratory tract, particularly the lungs, was long believed to be sterile. However, We now know it contains low-density, but metabolically active microbial communities. These microbes are influenced by aspiration from the oropharynx and may play roles in diseases like asthma, COPD, and COVID-19 related dysbiosis. Microbial balance in the oral respiratory region is crucial not just for local health, but also for systemic outcomes, such as cardiovascular disease, which has been linked to chronic periodontal inflammation. Let's now look at the urogenital microbiota, with a special focus on the vaginal microbiome, which is one of the most well-defined and clinically important microbial communities in the body. In healthy, reproductive-age women, the vaginal microbiota is typically dominated by lactobacillus species, especially L. crispatus, L. jensenii, and L. gasseri. These bacteria produce lactic acid, hydrogen peroxide, and bacteriocins, which help maintain a low vaginal pH, 3.5 for 0.5, creating a hostile environment for pathogenic microbes. This acidic environment is protective. It lowers the risk of bacterial vaginosis, BV, yeast infections, urinary tract infections, UTIs, and even sexually transmitted infections, STIs, like HIV. In contrast, when lactobacillus dominance is lost, a condition called dysbiosis, anaerobic bacteria such as Gardnerella vaginalis, Adipobium, and Prevotella may proliferate, disrupting mucosal immunity. The vaginal microbiota is dynamic and shaped by menstrual cycle, sexual activity, contraceptive use, pregnancy, antibiotics, and hormonal changes during menopause. For example, estrogen promotes glycogen deposition in vaginal epithelial cells, which supports lactobacillus growth. In men, the urethral microbiota is less studied but includes skin-associated genera like Corynebacterium and Staphylococcus. In both sexes, the urinary tract, previously thought sterile, contains a low-density microbiota, especially in the bladder, that may play a role in urinary health. Emerging therapies, like probiotic vaginal suppositories, aim to restore microbial balance and prevent recurrent infections. Understanding this ecosystem is vital for reproductive health, obstetrics, and urological care. Now that we've explored the major body sites, let's dive into what the microbiome actually does. Far from being passive passengers, these microbes are active symbionts that perform essential physiological roles. First, microbes help digest complex carbohydrates, especially those we can't digest ourselves, like dietary fibers and resistant starches. In the gut, this fermentation process produces short-chain fatty acids, SCFAs, like butyrate, acetate, and propionate. These metabolites nourish colon cells, regulate immune function, and influence lipid and glucose metabolism. Second, the microbiota is critical in immune system development and modulation. From birth, microbes help educate immune cells, promoting tolerance to beneficial organisms while remaining vigilant against pathogens. Germ-free mice, for instance, show underdeveloped immune systems. Third, the microbiota provides colonization resistance, occupying ecological niches and producing antimicrobial compounds that suppress invading pathogens. This is especially important in the skin, gut, and urogenital tract. Another fascinating role is in vitamin synthesis. Gut microbes produce vitamins B12, K, biotin, folate, and others that the host absorbs. Additionally, microbes participate in drug metabolism. They can activate, deactivate, or even toxify medications. For example, Egerthella lenta can inactivate the heart drug digoxin. This interaction field, pharmacomicrobiomics, is critical in precision medicine. Finally, the microbiome communicates with distant organs via the gut-brain, gut-liver, and gut-skin axes, influencing mood, cognition, inflammation, and disease risk. In short, the microbiome is a vital metabolic, immunological, and signaling organ, one that we're only just beginning to understand. Now, let's explore dysbiosis, a term used to describe an imbalance or disruption in the normal microbiota. Unlike a transient shift, dysbiosis reflects the loss of microbial diversity, an overgrowth of harmful microbes, or a reduction in beneficial ones. It can occur at any body site, but the gut is most frequently studied. So, what causes dysbiosis? Several factors contribute. Antibiotic use is a major driver. While life-saving, antibiotics can wipe out beneficial microbes along with pathogens, allowing opportunistic organisms to take over. Diet plays a huge role. High-fat, low-fiber Western diets are linked to reduced microbial diversity, whereas plant-rich, fiber-heavy diets support a healthier microbiome. Stress, infections, aging, pollutants, and chronic inflammation also influence microbial composition. Cesarean section births, formula feeding, and lack of early microbial exposure can predispose individuals to dysbiosis later in life. What are the consequences? Dysbiosis has been associated with a wide range of diseases. In the gut, dysbiosis is linked to inflammatory bowel disease, IBD, irritable bowel syndrome, IBS, colorectal cancer, and C. difficile infections. In the vaginal tract, it increases risk of bacterial vaginosis, preterm birth, and STIs. On the skin, dysbiosis contributes to eczema, acne, and psoriasis. Even systemic conditions like obesity, diabetes, asthma, and neurological disorders, e.g., depression, autism, have been linked to microbial imbalance. Importantly, dysbiosis may not be the cause in all cases. It can also be a consequence of disease. But it often contributes to a vicious cycle where microbial imbalance worsens inflammation, which in turn deepens the dysbiosis. In short, maintaining microbial balance is crucial for health, and its disruption can have wide-reaching effects. One of the most profound and intricate relationships we know of is that between the microbiome and the immune system. This interaction begins at birth and continues throughout life, shaping how the body distinguishes friend from foe. At the gut level, commensal microbes help educate the immune system. They influence the development of gut-associated lymphoid tissue, GALT, induce regulatory T cells, TREGs, and promote immune tolerance to non-harmful antigens, such as food proteins and beneficial microbes. This prevents chronic inflammation and unnecessary immune activation. Key microbial metabolites, such as short-chain fatty acids, SCFAs, especially butyrate, play a direct role. SCFAs act as signaling molecules that influence the maturation and function of immune cells like macrophages, dendritic cells, and T cells. Butyrate, for instance, promotes TREG differentiation, supporting immune homeostasis and reducing autoimmune risk. Certain bacterial taxa have immune modulatory properties. For example, Bacteroides fragilis produces polysaccharide A, which activates anti-inflammatory pathways through TLR2 signaling. Clostridium clusters 4 and XIVA induce TREGs and protect against colitis in mice. On the flip side, dysbiosis can impair immune function. A reduction in microbial diversity is linked to autoimmune diseases like type 1 diabetes, multiple sclerosis, and IBD, and to allergies and asthma, especially in early childhood. This is the basis of the hygiene hypothesis, which proposes that reduced microbial exposure in modern environments may predispose individuals to immune dysregulation. Microbes also modulate systemic immunity, influencing responses to vaccines, infections, and even cancer immunotherapy. In essence, the microbiome doesn't just coexist with the immune system, it's a critical architect of it. Supporting a healthy microbiome may prove key in immune-related disease prevention and therapy. Let's now focus on the critical concept of dysbiosis, which refers to a disruption or imbalance in the microbial community that supports human health. This isn't just about having bad bacteria. It's about a shift away from a stable, functional ecosystem toward one that contributes to disease. Dysbiosis can manifest in 3 key ways. Loss of beneficial microbes. This is perhaps the most concerning form. When key commensals, such as Faecalibacterium prosonitzii in the gut or Lactobacillus in the vaginal tract, are depleted, the protective functions they perform, like immune modulation, barrier support, or pathogen inhibition, are also lost. Overgrowth of pathobionts. These are microbes that normally coexist harmlessly within us, but become problematic when they proliferate. For example, Clostridium difficile in the gut or Staphylococcus aureus on the skin can cause serious disease if the competitive balance is disturbed. Reduced microbial diversity. Diversity is a hallmark of a resilient microbial community. When diversity declines, the system becomes more vulnerable to pathogens and less capable of adapting to environmental changes. This is often observed in people with inflammatory bowel disease, obesity, and after antibiotic use. What causes dysbiosis? A range of modern lifestyle and medical factors. Antibiotics are one of the most potent disruptors, especially broad-spectrum types that wipe out large swaths of beneficial microbes. Infections, particularly viral gastroenteritis or sexually transmitted infections, can rapidly alter microbial communities. Diet, especially low-fiber, high-sugar Western diets, starves beneficial microbes and promotes growth of inflammatory species. Stress and chronic inflammation also change gut physiology and, in turn, microbial composition. Dysbiosis is not always the root cause of disease. It can also be a result. But either way, it contributes to a vicious cycle of inflammation and immune dysfunction. Let's take a closer look at inflammatory bowel disease, or IBD, as a real-world example of how dysbiosis contributes to pathology. IBD includes Crohn's disease and ulcerative colitis, both marked by chronic inflammation of the gastrointestinal tract. While the precise causes are multifactorial, genetics, environment, and immunity, the gut microbiome is central to both disease onset and progression. In healthy individuals, the gut hosts a balanced microbial ecosystem that maintains immune tolerance and gut barrier integrity. But in IBD patients, we consistently see reduced microbial diversity, especially in firmicutes such as Faecalibacterium prosnitziae, a butyrate-producing bacterium with anti-inflammatory effects. At the same time, there's often an increase in proteobacteria, including adherent invasive Escherichia coli, which can penetrate the mucosal layer and trigger inflammation. This imbalance leads to impaired production of short-chain fatty acids, weakened epithelial barrier function, and excessive immune activation. Studies using germ-free mice have shown that colitis doesn't develop unless these mice are colonized with certain dysbiotic communities, evidence that the microbiota can drive disease in a genetically susceptible host. What makes this clinically important is that the microbiome is modifiable. For instance, fecal microbiota transplantation, FMT, has shown success in some IBD patients, helping restore microbial balance and reduce inflammation. Probiotics such as E. coli Nissl 1917 or VSL number 3 may help maintain remission, though results vary. Diet-based therapies, like the specific carbohydrate diet or exclusive enteral nutrition, can shift the microbiota in beneficial ways. So, IBD offers a compelling case study of how disruption in the gut microbiota contributes to chronic disease and how restoring balance can support treatment. Obesity may seem like a purely metabolic issue, but we now know the gut microbiome plays a significant role in energy balance and fat accumulation. Dysbiosis doesn't just reflect obesity, it can help drive it. One of the most consistently observed changes is a shift in the firmicutes to bacteroidetes ratio. In both mice and humans, a higher proportion of firmicutes and a reduction in bacteroidetes has been linked to increased caloric extraction from food. This means that the same meal can yield more usable energy, depending on the microbial profile. In addition to energy harvest, gut microbes influence fat storage, glucose metabolism, and appetite regulation through various signaling molecules and metabolites. One key player here is lipopolysaccharide, LPS, a pro-inflammatory molecule produced by gram-negative bacteria. In dysbiosis, increased LPS can cross a leaky gut barrier, triggering chronic low-grade inflammation, a hallmark of obesity and insulin resistance. Animal studies have made this even clearer. When germ-free mice receive fecal transplants from obese donors, they gain significantly more weight than mice receiving microbiota from lean donors, even when their diets are identical. This confirms a causal relationship between microbiota and metabolic phenotype. Several external factors contribute to obesity-related dysbiosis. Western diets high in fat, sugar, and low in fiber. Sedentary lifestyles. Repeated antibiotic exposure early in life. Therapies that aim to reshape the microbiome, such as high-fiber diets, polyphenol-rich foods, prebiotics, and targeted probiotics, are currently being tested as adjuncts to weight management. So, while obesity is multifactorial, the microbiome adds a critical, modifiable layer, one that could unlock new treatment and prevention strategies in the future. Beyond gut health and obesity, the microbiome is now linked to a broad spectrum of systemic diseases, many of which were once thought to be unrelated to microbes. These links are not just correlations, there's growing evidence of causal and mechanistic roles. Let's start with allergies and asthma. The hygiene hypothesis suggests that reduced microbial exposure early in life may lead to improper immune development. Children born via C-section, formula-fed, or raised in ultra-clean environments often lack microbial diversity and have higher rates of eczema, asthma, and food allergies. Specific microbes like Bifidobacterium infantis help train the immune system to tolerate harmless antigens. Next, type 2 diabetes. Studies consistently show that individuals with this have reduced gut microbial diversity, along with lower levels of beneficial butyrate-producing bacteria and higher levels of pro-inflammatory species. Dysbiosis may exacerbate insulin resistance via chronic inflammation, altered bile acid metabolism, and impaired gut barrier function. Then there's the gut-brain axis, one of the most fascinating frontiers. The microbiome communicates with the brain through immune, neural, and hormonal pathways. In autism spectrum disorders, researchers have observed altered microbial profiles, often accompanied by GI symptoms, and some early trials suggest that microbiota modulation might improve behavioral symptoms. Similarly, in Parkinson's disease, patients often show gut dysbiosis years before motor symptoms appear. Inflammation and microbial metabolites may influence the vagus nerve and central nervous system. Even depression is now linked to microbial imbalance. What some are calling psychobiotics are being explored as treatments to boost mood and cognitive function via microbial metabolites like GABA and serotonin precursors. So, the microbiome is emerging as a unifying factor across diseases, connecting the gut to the immune system, metabolism, and the brain. Given the microbiome's profound influence on health, the natural next question is, can we change it to prevent or treat disease? The answer is yes, through several promising strategies that aim to restore balance and enhance beneficial microbial functions. First, probiotics. These are live beneficial microorganisms, often from genera like Lactobacillus, Bifidobacterium, or Saccharomyces. They're available as supplements or in fermented foods like yogurt, kefir, and kimchi. Probiotics can help repopulate the gut, outcompete harmful microbes, and modulate immunity. While some strains are effective in specific conditions, like Lactobacillus rhamnosus for antibiotic-associated diarrhea, results can be inconsistent, and strain specificity is key. Next, prebiotics. Unlike probiotics, these are non-digestible fibers such as inulin or fructooligosaccharides that serve as fuel for beneficial microbes. Prebiotics selectively promote the growth of butyrate-producing bacteria, which strengthen the gut barrier and reduce inflammation. A diet rich in diverse fibers is one of the best long-term ways to support a healthy microbiome. Then we have fecal microbiota transplantation. This involves transferring processed stool from a healthy donor into a recipient's gut, typically via colonoscopy, enema, or capsules. FMT is now an approved treatment for recurrent Clostridioides difficile infections, with cure rates over 85%. Research is expanding into its use for IBD, metabolic diseases, and even neurological conditions. Lastly, diet is the foundation. High-fiber, plant-rich diets provide a constant supply of prebiotics. Fermented foods like sauerkraut, miso, and kombucha introduce beneficial microbes. In contrast, Western diets high in fat, sugar, and processed foods reduce diversity and promote inflammation. Ultimately, modulating the microbiome isn't about a single pill, it's about sustained, ecosystem-level change. And that starts with daily choices in what we eat. Let's end this section with a real-world success story that showcases how powerful microbiome-based therapies can be. This is the case of fecal microbiota transplantation, or FMT, used to treat recurrent Clostridioides difficile infection, commonly known as C. difficile. C. Difficile is a spore-forming bacterium that can overgrow in the colon after antibiotic treatment. Antibiotics often kill off not only the pathogen but also the protective gut flora, leaving a niche for C. difficile to thrive. The result is severe diarrhea, colitis, and, in recurrent cases, significant morbidity and even mortality. Traditional antibiotic treatments like vancomycin or fidaxomicin often fail in recurrent cases, as they don't restore the microbiome's diversity or competitive structure. That's where FMT comes in. The principle is simple but profound. Transfer stool from a healthy donor, containing thousands of species of commensal microbes, into the patient's colon. This reintroduces a stable, diverse microbial community that can outcompete C. difficile and restore normal gut function. The results have been remarkable. Clinical trials show over 90% success rates in resolving recurrent C. difficile infections, often after just 1 or 2 transplants. This is far more effective than antibiotics alone, and without the long-term disruption to the microbiome. FMT can be administered via colonoscopy, enema, nasoenteric tube, or even in capsule form. Safety protocols are strict. Donors are screened rigorously for pathogens, and samples are processed under sterile conditions. This case marks a paradigm shift in medicine. Instead of fighting pathogens with drugs alone, we're now restoring ecosystems. It's a model that may one day extend to many other conditions, from IBD to neurodegeneration. So, FMT for C. difficile is more than a treatment. It's proof of concept that microbiome-based therapies can work safely and effectively. Let's end this section with a real-world success story that showcases how powerful microbiome-based therapies can be. This is the case of fecal microbiota transplantation, or FMT, used to treat recurrent Clostridioides difficile infection, commonly known as C. difficile. C. difficile is a spore-forming bacterium that can overgrow in the colon after antibiotic treatment. Antibiotics often kill off not only the pathogen but also the protective gut flora, leaving a niche for C. difficile to thrive. The result is severe diarrhea, colitis, and, in recurrent cases, significant morbidity and even mortality. Traditional antibiotic treatments like vancomycin or fidaxomicin often fail in recurrent cases, as they don't restore the microbiome's diversity or competitive structure. That's where FMT comes in. The principle is simple but profound. Transfer stool from a healthy donor, containing thousands of species of commensal microbes, into the patient's colon. This reintroduces a stable, diverse microbial community that can outcompete C. difficile and restore normal gut function. The results have been remarkable. Clinical trials show over 90% success rates in resolving recurrent C. difficile infections, often after just 1 or 2 transplants. This is far more effective than antibiotics alone, and without the long-term disruption to the microbiome. FMT can be administered via colonoscopy, enema, nasoenteric tube, or even in capsule form. Safety protocols are strict. Donors are screened rigorously for pathogens, and samples are processed under sterile conditions. This case marks a paradigm shift in medicine. Instead of fighting pathogens with drugs alone, we're now restoring ecosystems. It's a model that may one day extend to many other conditions, from IBD to neurodegeneration. So, FMT for C. difficile is more than a treatment. It's proof of concept that microbiome-based therapies can work safely and effectively.